Making a wet mount. A wet mount is normally done with eukaryotic cells such as pond water or human blood. It can be done with dry materials such as fungal cultures or a scraping of tissue. The concept is to place the material in water or a liquid dye that can then be observed. Start with a clean microscope slide. Place a drop of water or stain. We can use many types of samples. This material here is a fungus that was growing on a croissant that was left over. A little bit goes a long way. I'm just going to grab a little bit of the fungus. I put it in this drop of water. Then we take the cover slip. Since we are dealing with a wet mount, we do not want to put this in the microscope without having protection for the lens. Gather up our water, then allow the cover slip to go down. Very gently touch this. The concept here is to flatten this out. As we flatten it, that will reduce the thickness of the sample. As it reduces the thickness of the sample, it will increase our ability to observe. If we need to add a little color, we can add a drop of color to the edge of the slide. You can see the color migrating under the cover slip. If you want to give it a little more help in doing that, there are two ways to do that. The first, place a piece of paper towel or toilet paper on the other side of the cover slip. It will draw the water across. You can see that the dye actually does migrate. Then just give it one or two very slight taps. It will be ready to be observed. If there is excess moisture on the outside, I can blot this and that draws the cover slip down. I now have a wet mount of material that I can look at under the microscope. The other way is you can pick up the edge of the cover slip with a needle. Sometimes this is a little bit tricky because you don't want to break the cover slip. Place a drop of dye, then lower the cover slip back. You also don't want to leave these air bubbles on the inside. We apply a little bit of pressure and you can see the air bubbles squeeze out. Now we've got the material on the inside, and then you can blot the edges. You can take this and you can observe it. Making a wet mount is a very common way to look at fresh specimens. There are many variations on the technique, but the same basic principles apply.